Thank you very much all for coming for our webinar today. The biophotonics technologies against COVID. I aim to convince you that biophotonics technologies have contributed to several applications during the pandemic. And these applications will go from remote disease monitoring, home healthcare, telehealth, mobile health, and especially mobile health, home healthcare was especially important given that we couldn't meet our doctors in person. Biophotonics technologies were also automated and made users independent on monitoring their vital signs and health. Biophotonics has also been used for surface disinfection and decontamination. And also the technologies and applications which have been researched are useful for detecting, monitoring, treating diseases other than COVID. So it will continuously contribute to advances in healthcare with biophotonics. So if you think about the context of COVID, you might know at this point that the transmission routes will be mostly divided into two. One is the airborne route. This is mostly based in the droplets, which will be released in the air by the sneezing and coughing from infected individuals. And these droplets can travel to a healthy individual, infecting him or her. So the transmission can also occur with contact routes. This could be either a direct contact with an infected COVID patient or an indirect contact with infected surfaces. And due to the contagiousness and the virulence of SARS-CoV-2, so the virus which causes COVID-19, the World Health Organization has issued a guidance for infection and prevention and control strategies, which involves the social distancing and the use of PPE that you are all used to at this point. Now, if you think about how people have dealt with the COVID outbreak, you would look at not only the use of PPE when you go to a supermarket for your grocery shopping or any stores in your everyday life. You would look at the use of PPE by physicians in their work so they can protect themselves against COVID patients and any infected material that could be in there in the surfaces. We would have during the outbreak, we would have at this steadily increasing number of cases, and due to this, there was a shortage of these four components here. So mostly the PPEs, the physicians, the hospital beds, and negative pressure rooms, as known as isolation rooms as well. So due to this, we had a number of technologies created by biophotonics which helped to solve some of those problems. So you would have the contamination of surfaces in hospital rooms. You would have actually the telehealth helping the clinicians to give the best recommendations for their patients. You would have some of the decontamination technologies used even for helping people to reuse their PPEs where there was no enough supply for medical staff to use new PPE. And we will discuss about all these today. Now, if you think about medical procedures, you would see also some of the procedures which needed caution. So if you think about the ones that use biophotonics technologies such as light therapy, skin treatments, endoscopy, colonoscopy, surgery, dental procedures, you would think about all the procedures who would actually have a chance to harm medical staff due to the contact with infected materials such as the biofluids of a COVID patient. And these actually needed to be taken with extreme caution. Some of them would not be performed unless in extreme cases, and also the facilities were actually renovated so that there was one directional airflow, and also the safe distance could be obeyed at all times. Now, not only the medical procedures were changed, but also the medical examinations. So you can just think about some of the conventional medical examinations you would have, just measuring the blood pressure with a cuff, just examining the ears with an otoscope, your temperature with a normal thermometer. And also, you wouldn't be able to meet your doctors in person anymore. So medical appointments wouldn't exist. In this case, biophotonics technologies have contributed to some of those examinations. Um, there are some biophotonics technologies which are used to measure blood pressure and respiratory rates, or even temperature from a distance. But here we will mostly focus on the infrared imaging, which is used for measuring temperature from a distance. It is the same technology you can find in thermometers. And you would also see some, we also discussed about pulsoximetry, which could be used to monitor the blood oxygenation, especially in patients with post-distress, pulmonary distress caused by COVID. And if you think about all these that we discussed today, we will think about how the in-person medical appointments were made before, and now with COVID, they were not possible. In that context, the actual monitoring of patient vital signs was no longer an ideal, it was a requirement. And therefore, biophotonics technologies needed to be developed for those. There are a number of contributions of biophotonics to home healthcare and telemedicine due to this monitoring of patient vital signs. You could think about just any smartwatch, any wearables that you may have contact with, you may have used for monitoring your performance during exercises or sports at home. You would see how you could monitor your sleep quality. You could actually monitor some of your vital signs with your smartphone technologies and apps. So all these are already biophotonics technologies that you already apply in your daily life, and some of the doctors could actually talk to the patients and base some of their conclusions and recommendations on what the patients observe in terms of vital signs, looking at the readings of certain equipment. Now, if we actually want to have accurate readings for medical use, it is not so simple to use just the wearables and smartphone tech. That if you want to have precision to actually read any biological information with um, with the precision of for medical use, then you would have to have medical equipment, extremely high quality medical equipment. And in this case, we are looking for technologies where medical research actually takes about 13 years from going from bench to bedside. During the COVID outbreak, it was a nightmare to actually get new technologies applied in the clinic because many of the companies were actually closing business. So how we actually generate new technology to put in use in the clinic during the pandemic situation. So what's happened is that several institutions such as the European Commission, the NIH in US and many others worldwide actually incentivized the development of technologies against COVID. And this was either financially making the law more flexible for research ethics approvals and commercialization. You would see several projects that were started because of this. I listed here just a few of them in the right side. But I emphasize that there were many others that were actually started due to this incentive. And one that I would actually highlight here is the COVID home test by Illumi. These are based on fluorescent quantum dots, and the company actually was planning to close after 3 months from the start of the pandemic. And actually with the program, with the financial incentive and so on, they actually produced COVID home test they went from research to product in about 9 months. So starting from April, and they actually launched the product on December. So you would see how quick technology can actually be developed if there are no barriers, if there is a collaboration between several institutions, if there is multidisciplinary knowledge actually with collaborations with many institutions. It can be very promising to actually just solve daily life problems. And we could see this happening during the pandemic. The technological advancements were very, very sped up. They were very accelerated. And we discussed some of those just last week during the European conferences on biomedical optics, especially on the COVID panel discussion, where our BioPhotonics team played a key role there on the organization, the advisory committee, and also on the questions to the panel. So on the COVID tests, we know that BioPhotonics has contributed to a number of those. It's not only at home, but even if you think about the conventional COVID tests, you would see something like this here. So you would just collect either a sample of mucus from the nostril or throat swab, or sample of saliva, and you send the sample with the cells that might be infected by COVID to an analysis, which is the RT-PCR, so the reverse transcriptase polymerase chain reaction. And in this case, in the first part, we just convert the virus genetic material, the RNA, to DNA, and this DNA is actually amplified. It can be amplified 1 billion times in an hour, for example. And with the sample with the DNA amplified, there is an addition of a fluorescent dye to actually dye the viral genetic material. And what we do is we just detect the intensity of the fluorescence so that we know how many copies of the virus we can find in the sample. And an infected individual will just have a higher number of the copies of genetic material of the COVID virus. This is how the COVID tests work. And now you would see that not everyone will do COVID tests and will not do them every time. It is a kind of uncomfortable test to be done. It also takes some hours to be done. So we would need to narrow down the population that will do COVID tests. We also need to look for when this test needs to be performed. And to do this, we can use infrared imaging. So you would be looking mostly to the fever symptoms in case there is a higher temperature reading than usual. And this technology is present in just thermometers, infrared thermometers. You could have a point measurement and in several locations of your body, or you could just read the temperature profile in several parts at the same time, and even in many people simultaneously. The infrared technology is actually based on the black body radiation, which is the light that we emit due to the vibration of our molecules, which is basically temperature. So if you want to have an idea why we emit light, basically everything emits light when it's heated. We just don't see it. So think about the blacksmith that you see in movies when they are actually forging a sword. So in the beginning, the sword doesn't glow, but if you heat it in the forge, you see that it becomes red. If you heat it even more, it becomes even yellow. And if you could heat it to the temperature of the sun, it would appear like white. That's why the light of the sun is white. It has a high temperature, and that's why it emits light. Infrared cameras would detect infrared radiation around this region here, and this is how we look at the intensity and we measure temperature this way. The infrared sensors would be mostly based on bolometer sensor technology, and these would be divided into 3 types. You could have semiconductor technology where the radiation is converted into current. You could have a technology based on thermoelectric effect where the radiation is converted into voltage, or the paraelectric effect where the radiation is converted into resistance. And in all these technologies, actually, the sensor can be very miniaturized, so you can have compact infrared cameras. And these chips are pretty much similar to chips that we produce in Tyndall, for example. You would see these sensors being produced with CMOS fabrication process with silicon and vanadium oxide. Each of the pixels of the camera would look like a unit like this with a microbolometer array. You would have just an infrared absorber layer at the top. It is followed by a reflective layer on the bottom, separated by a vacuum, layer of vacuum, and underneath there would be a readout circuit. If you look at this structure on the side, you would see that there is a high vacuum in the middle between the infrared absorber layer and the reflective layer, so that we don't actually have heat loss during the measurement of temperature, and we can have an accurate measurement. If we want to have measurements really accurate, we need to take into account the temperature variations in the environment, and we will look at having more accurate measurements in, for example, acclimatized rooms or isolation rooms, where you can actually control the heat exchange rates and also control the airflow there. Still, we have this applied in, for example, entrances of buildings. If you went to Tyndall, you see a camera there. You can look at your temperature reading before you enter and know that you are not suspect for COVID or having any fever or anything like that. Now, the infrared imaging technology has proposed to be used in crowded places, especially for identifying then the potential people to do COVID tests. And this could be actually coupled to face recognition and reporting. But there are privacy issues which hinder the application of all these technologies together. So it wasn't done. It could be done in extreme cases, but it was just a proposed application. Still, it is a very useful technology. And another technology that we could see during the pandemic was the pulsoximetry. In this case, we are looking at the blood oxygen saturation ranges. You could see that for a healthy individual, we would actually have more than 90% of oxygen saturation. If the oxygen actually decays, the oxygen readings decay to about 90%, we would identify a situation of pulmonary distress. And with people with acute illnesses, you would see even readings even below 90%. If the readings, the blood oxygen saturation actually drops to below 80%, we could have the patients with impaired mental functioning. And also it is very dangerous for the patients if we are not able to act fast. And continuously monitoring the blood oxygen saturation is very important. Not only this, it is important to monitor these readings very accurately. So we would look mostly for readings of finger pulse oximetry, which meets the quality standards of this ISO norm that I wrote here. And there are several conditions you can actually take the readings. You would need a warm finger, the patient sitting upright, you need to stabilize the reading for a minute before considering it right. You could have mostly low readings if the patient has some conditions. You could and you, as we were talking about before, we can't use wearables that weren't designed for medical use because they are not as precise yet. So for example, we we wouldn't use a pulse oximeter for this reading. Like, could I use a smartwatch for taking the blood oxygenation of a COVID patient? No. Could I use just, could I put a Netflix socks, my Netflix socks on the COVID-19 patients and let him or her just watch a movie and very happy just monitor their blood oxygenation and see what is happening? No. But if you want to actually create a technology which could make the wearable sensors, the miniaturized sensors, very precise for medical use, please join us. And actually, you might, if you actually create this type of technology, maybe you earn a Nobel or you'll be rich. Who knows? Why not? You would you would have this opportunity. So postoximetry has been very promising. It needs to have a bit more precision, and it will also perform. It will have a key role in other technologies as well, not only in COVID. Now, if you think about other another technology, which is the surface disinfection and decontamination, as we saw before, um, we had this type of technology already applied for several decades in hospital rooms and even to decontaminate the surface of food in food industry. And now in the pandemic, we had this technology booming now that we need environmental surface disinfection everywhere, especially in crowded places such as supermarkets, airports, post offices, and even in industries that couldn't stop during the pandemic, such as the pharmaceutical industry and the food. These industries just can't stop. You can imagine just living just without any food and your medicines. So um, the surface disinfection technologies are based on the ultraviolet light. They were also used for decontaminating respirators for their reuse. You would have, in case it is absolutely necessary, if there is no supply for PPE, there are people who have used this to decontaminate the masks, respirators. And now we would have several types of the ultraviolet germicidal radiation or UVGI equipment that you can make. You can use light emitting diodes or LEDs or UVGI lamps. And LEDs will be more compact. They are more versatile. You can make equipment of different shapes, smaller. You can store them almost everywhere, which was really good for hospitals at the time when they were crowded. There was no space to store anything. They are bright, and there are several advantages to them. Now, lamps, they are even brighter. They will actually be able to be used to decontaminate many masks, many respirators at a time. They have a standard model as industry standard. And there are several ways of using each of those technologies. Some of the parameters are, for example, the contamination time, which is very variable depending on the application and which technology you use. This goes from 30 seconds to 30 minutes. Especially for lamps, you would have a power that is about 10 watts, and for lamps you would use UVC on the 254 nanometer range. In this case, we have the production of ozone, which is toxic. So if we are using lamps, we need to minimize breathing close to the equipment which is using UVC. And also we need obviously to be careful to not expose our bare skin or eyes to UV radiation. And here are just some of the advantages and disadvantages of LEDs and lamps. They can both be used to decontaminate masks from illumination at different angles, which is more efficient than. These technologies have been used in multiple types of equipment. In low resource settings, people were actually very creative. You would see sterilization boxes. For people who couldn't buy a new equipment, you would see people actually modifying the biosafety cabinets of that emits UVC, and they would just use those for decontaminating some of the masks. Now UVC is actually affects the electrostatic charges of the masks, so of the propylene material in the masks, and this actually compromises the respiratory integrity. So it's not so ideal if actually you eliminate the mask for a lot of time, or if you deliver a lot of light dose. So some of the researchers actually looked into the decontamination of these PPE with UVB instead, which would be in another region between 280 and 315. In this case, they would be using some of the light therapy boxes of psoriasis, and that is actually present in almost every dermatology office. Now, all these technologies could be used for surface decontamination and decontamination of PPE. What needs to be actually done is actually the protocol needs to be fixed so that the irradiation time and the light dose actually doesn't provides good decontamination without damaging the respirator. So this is the most important point. And there were several protocols that were developed for that. Now, we have seen some of the technologies that were used during the pandemic. There were some of them that were also proposed and researched. Here I listed some few, just a few of them, especially those which we didn't see in the previous webinars. So we can look at some of the different content here. And those would be just the Fourier transform infrared spectroscopy, so as known as FTIR, the Raman spectroscopy, NearInfrared spectroscopy, which could be combined with photodynamic therapy. And let's have a look at the FTIR. So this is a type of vibrational spectroscopy, which is related to the vibration of the chemical bonds of the molecules in the sample. And because it is so, because it is dependent on the vibration of the molecular bonds, it is very molecular specific. So you could see signals from, for example, the genetic material, the DNA or RNA in the sample. You could see proteins that could be on the capsule of the virus, and that's why it has potential to detect COVID in several types of samples, such as biofluids. Could be saliva, could be blood, or any other type of biofluid. And what you would be looking for is a reading like this. You would be looking for a difference in reading from a normal patient and a COVID patient. So the FTIR technology is a very mature technology already, because it has been developed in the past century. The first equipment was actually were actually created on the 1950s, and you would currently see that spectrometers are very portable and reasonably cheap. They would require just a minimal amount of sample for analysis, and the analysis can be done in real time. So some of this analysis can be also empowered by looking at microfluidic chips. You could have any analysis on fluids that you just insert in a small amount in these chips, and these chips could be actually sent everywhere. You could have high throughput screening in crowded places, for example, airports. You could actually have an analysis, a quick analysis just before letting someone enter the airplane, for example. And if this technology is actually automated, then there would be no operational costs. So it was a very promising technique to be applied for COVID detection. Another technology would be the Raman spectroscopy. And in this case, we are looking at another vibrational spectroscopy technique. So very, very molecular specific as FTIR. And the advantage of Raman compared to FTIR is that it provides water-free signals. So we are not affected by the water in the sample. And for example, we could use this to target biomarkers in cells infected by the SARS-CoV-2. So we we would see if we have infected cells, we could monitor the progression of the cell and the biochemical profile of this evolution of, of the infection, so that we could see how it evolves in terms of lipids, protein, nucleic acids, and actually determine the molecular origin of the disease. If we are able to identify which type of biomarker could be used to identify COVID earlier, or also look at the biochemical profile to actually propose new treatments for COVID, this would be a very useful technology which would not only be used to look at the molecular origin of the disease, but also to propose several other technologies to be applied to it. Now, Raman spectroscopy can also be used in a very different angle. It could be used to analyze the volatile organic compounds in your breath. So you could have a breath analysis. And these compounds are actually gases that you release when you just breathe. And since COVID is pulmonary, leads to respiratory problems, you would see a lot of things happening in the lungs. And some of the gases that would be released could be different in COVID patients. So the proposition was that if we have a sensor where you have nanoparticles on the surface, these nanoparticles can actually amplify the signal from volatile organic compounds that are attached in the surface of the sensor. So you have someone breathing in the sensor and you detect these organic compounds, these gases, in a very amplified signal. And this is called surface enhanced Raman spectroscopy. You could have this technology applied in a lab-on-a-chip device, and this could be incorporated in a respirator, for example, so that the gas actually can accumulate on the surface of the sensor, and we could actually have this this type of detection quicker and in an earlier stage. Now, also related to the pulmonary distress caused by COVID, we would have several critical conditions with, for example, the acute respiratory distress syndrome. In this case, we will have raised permeability of the capillaries of alveoli. And with this increase in permeability, there is a leakage of fluids inside the alveoli. And this actually damages it and it doesn't let it to uptake oxygen. So that's why the oxygenation of COVID patients actually drops. And we can actually look at this leakage of fluids with near-infrared spectroscopy. So since it is a fluid that is rich in proteins and water, we could detect this type of fluid accumulation with near-infrared spectroscopy, which is very sensitive to water signals and protein signals, such as those you can see in the figure here. Also, by using near-infrared, then we can look at deeper tissue layers, because if you remember, the near-infrared is in the optical window that we have seen in the first webinar, and this is the window where we are most transparent to light. This is the wavelength range where light will be mostly transmitted through the tissue. And with this, we can also simultaneously take readings of blood oxygenation in deep tissue layers. We can use both information of fluid accumulation and blood oxygenation to actually monitor the recovery of COVID patients. We can monitor their treatments, we can monitor how they recover, and if they actually recover all the functions well before they get actually, before they go out of the hospital. So not only for disease detection and monitoring, but near-infrared spectroscopy could be combined with treatment. So we could have theranostics applications, which is theranostics is a combination of therapy and diagnostics. We could use the near-infrared lights to actually do extracorporeal illumination, as you can see here, and do antimicrobial photodynamic therapy. If you remember about the photodynamic therapy that we have discussed in the first webinar, it is dependent on making what you want more sensitive to light and actually killing that organism. So in this case, we would need a type of photosensitizer which accumulates preferentially in the viruses, in the COVID virus. And the way it works is that the photosensitizer could be attached to the virus, mostly, if possible, on the genetic material or the capsule. And then once the photosensitizer is illuminated, it can be excited, and in the presence of oxygen, this photosensitizer can actually transfer its energy to oxygen, and it can lead to the formation of singlet oxygen or reactive oxygen species. And these types of species will actually react with almost anything around, especially damaging than the virus genetic material, so the virus RNA. And with this, the virus wouldn't be able to proliferate anymore. So that's how we would be able to treat, and also to treat at the same time we can detect and monitor the treatment and the condition of COVID patients. So now I hope I have discussed some of the technologies here, and I hope I have convinced you that biophotonics has contributed to several applications during the pandemic, including remote disease monitoring, home healthcare, and also with the automation of several of the biophotonics technologies, users have were independent to monitor their vital signs and also their health during the pandemic. We could have a number of surface disinfection and decontamination technologies, and the technologies and applications researched and developed are useful to detect, monitor, and treat diseases other than COVID, so we can advance healthcare in that way. So here are just some references. If you want to just contact me, you can use these links. You can have this presentation afterwards. And if you want to know some of the proposed technology, I just leave here some of the proposed papers that I would outline some of the technologies we have discussed here.